if I can get my screen to advance, which it is. Hold on just one sec. Okay, great. So today we're going to cover a few things. I'm going to talk briefly about a meeting at the FDA that occurred yesterday, just because it's fresh on my mind. We'll talk about the new grants that we funded for 2016, the second round of grants that we funded, and I'm going to talk about two different research advances in PWS that are have just occurred and I think are of interest to our community. So to touch on the Commissioner's listening session, so I was quite surprised and pleased to get an invitation actually right before Thanksgiving to attend the FDA Commissioner's listening session. So apparently the Commissioner occasionally has meetings in which he invites industry or academicians or patient groups to discuss their priorities and concerns. And it's just, I guess, a way for him to get a pulse on what's going on in the in foods, drugs, and among patient groups and what is relevant to the FDA. And so it's the FDA leadership, which includes the Commissioner, who's the head of the FDA, as well as the directors of the Center for Drug Evaluation and Research. That's where most of the PWS drugs sort of go through as they're being evaluated for approval in PWS. CBER, which is the Center for Biologics, CDRH, which is the Center for Devices, so things like the vagus nerve stimulator and any transcranial direct current stimulation, which we also have development in PWS for. And then the Orphan Drugs Office, which focuses on drug development for rare diseases, which of course is relevant to us. And this particular listening session included rare diseases, as well as rheumatology and infectious disease. And I'm not sure how they grouped those together, but that's how they did it. So from the rare disease perspective, they had some groups that are overarching that address many aspects of rare disease, like the National Organization of Rare Disorders and Every Life Foundation, which focuses on improving the drug development process. And then several patient-specific organizations were invited, including the Cystic Fibrosis Foundation, Parent Project Muscular Dystrophy, Cure SMA, and a couple of others, and FPWR. So first, I thought it was really notable that you know, our community was invited to this because, as you know, there are thousands and thousands of rare diseases, and so it was really exciting to be invited, and it lets us know that we are on the FDA's radar and that the FDA is interested in learning more about our community because probably in at least in part, there's very active drug development in the PWS community. And so each organization was asked to bring their priorities and concerns for discussion. So one of the things that came up right away, because yesterday was the signing of the 21st Century Cures Act, there was a lot of discussion about that and how it will impact the FDA. So the Commissioner, after the meeting, was going to the White House for the signing ceremony, and this is a piece of legislation that's been in the works for at least 2 years, and I'm no expert in the 21st Century Cures Act, so I'll just say that. But there is a lot of information that's available online, and there are many aspects that will impact both research and drug development in the U.S. There's additional funds for the NIH. There is funds for the FDA to modernize some of the clinical trial design. And also, it really puts into place or formalizes the role of the patient voice in the drug development process. So really, it's just been over the past few years that the FDA has been making a concerted effort to bring the patient voice into the drug development process, and this will formalize that. It will provide more guidance for that, and it will really make it a pillar of the FDA's drug development process, which is very exciting for us. So I have at the bottom a couple of links if you want to learn more about how 21st Century Cures will impact the FDA and also how it will impact the National Institutes of Health, which supports basic science research in the U.S. But to get back to the concerns that we brought to the FDA, they're shown here. We listed some priorities for our community and also some concerns. The priorities included improving institutional knowledge of PWS at the agency. As we've said, there are thousands and thousands of rare disease, and it's a lot to expect that the reviewers at the FDA be really familiar with each of those. So we have a responsibility as a community to provide them with information about the unmet medical need, about the burden of disease, about what is important to families and individuals with PWS that will help them in making decisions, and also, what sort of risk we're willing to tolerate, what sort of side effects we would be willing to tolerate, for example, if a drug were effective against hyperphagia. And the Prader-Willi Syndrome Clinical Trials Consortium is addressing some of these issues and trying to gather data on all of those aspects. And so bringing that to the FDA in a manner that is efficient and that they can really, it can really be useful for them was one of the things that we brought up, and also was a concern of many of the other advocacy groups. A second priority was improving interaction and engagement with the FDA. It can sometimes be a confusing, if you've ever seen their organizational chart, it can be quite confusing. There's an office of orphan drugs, there's an office of health and constituent engagement, there's patient engagements within some of the centers, so it's a little challenging to navigate. And sometimes the interaction can be left to the very end point where a drug is seeking approval or not approval. And what we were suggesting, and again was echoed by many of the other advocacy groups, was that there be engagement throughout the drug development process. So very early on, so the FDA understands that what endpoints are important to the patients. We want to know how to work with the FDA to provide additional clarity about the FDA on the FDA's thinking on PWS drug development. And so for industry to find Prader-Willi syndrome attractive, they have to sort of know the rules of the game. What is the FDA looking for? What do they expect in a drug? And so if we can work with the FDA to provide some of that information to industry and to academic investigators, that will make our rare disease more attractive. And finally, a concern that we brought up and that was discussed at some length in a variety of settings was the perception of conflict of interest. In rare diseases, we're small communities. We as an advocacy group find it very important to engage with industry partners early and work with them to try to make the clinical trial process as efficient as possible. But that carries with it the potential for a perception of conflict. If we've been working closely with a particular pharmaceutical company, then the perception could be that that we're favoring one company over another, for example. The FDA also has to manage this. They want to work closely with patient groups, but they don't want to be seen as being biased by the patient groups. They want to maintain sort of a bit of a wall when it comes to analyzing scientific data. And the companies also have an issue that they have to manage with respect to conflict of interest. They want to engage with the patient organizations, but they don't want to be seen as trying to advertise to the patient organization. So there's a lot of potential problems, and I think the consensus was that these are tricky questions. They need to be managed with as much transparency as possible. But in a rare disease community where it's a very small community, these things they're going to come up, and so we're going to have to manage them. Some of the other takeaways from the discussion was the importance of patient registries. This came up in many aspects within the rheumatology group, within the rare diseases, of course, the importance of registries for monitoring patients over the lifespan. So traditionally, when a new drug comes on the market, the FDA asks that company to have a registry associated with that drug so they can see if there's, for example, side effects down the line. But actually everybody kind of agreed that it makes much more sense, especially for disorders that are across the lifespan of an individual, such as with PWS, if the registry is about the patient. So it's a patient-centered registry. And then whether they're moving on and off drugs, depending on what's coming up. You know, you can have information across the lifespan of the person about how effective drugs are, the safety profile of drugs, and also to gain a better understanding of the natural variation of any particular disorder. So all of those aspects are critical for understanding how well a drug is performing, understanding the medical needs of the community, and the FDA reiterated it, the need for good patient registries. As I mentioned a little bit already, a recurring theme was the challenges for patient groups navigating the offices of the FDA, and NORD, in fact, has suggested a reorganization of the FDA to include a specific office that manages all of those aspects. So we'll see if that moves forward. There was discussion on using biomarkers as endpoints in clinical trials. So, you know, if there, for example, the example in PWS might be if there were a blood test that would show if a person had high or low hyperphagia, how that might be a much better measure than a questionnaire. But in any disorder, you know, there's the potential for biomarkers, and there's a lot of scientific discussion about making, you know, how to develop good biomarkers. And I already mentioned the issues around conflict of interest and managing that in a way that we use the expertise of all of the stakeholders, but we don't have any bias from any of the stakeholders. So it was a really interesting morning, and it was really an honor to be there. And you know, there was, at this point, it was a lot of discussion and people expressing their opinions and perceptions. And I think the hard work will come in actually trying to implement some of these suggestions into action. But it was great to be there, and it was great to have PWS sort of front and center in front of the leaders of the FDA. So that was very exciting, I think, for the community. So I'll switch now to a discussion of the new grants that we have provided funding for recently. And just as a reminder, as I like to remind myself and all of us, the mission of FPWR is to eliminate the challenges of Prader-Willi syndrome through the advancement of research. And we do that in two ways. We have a general grants program, and that's what we'll be talking about today. And that's our support of investigators who bring their ideas to us. So they, you know, have ideas about, you know, how to advance our understanding of PWS, how to develop new therapies, and they, through a competitive grant process, we provide funding for the best ideas. I'll just make a note about, as we move into 2017, one of the things that we're working on, or we've developed, is a research plan, and you can, here's the link to our 5-year research plan. And what we'll be complementing that investigator-initiated research with in the coming years is a more directed research program where FPWR and the PWS community is actually identifying some of the tools and resources and programs that we feel are necessary to move the field forward, and providing support for that. So we feel it's important to be doing both. So the grants program is a great place for the most innovative research, the best ideas that investigators have, whereas the directed research programs are for moving forward efficiently in areas that we know that we want to move forward in. So we'll have other webinars discussing in more detail about the directed research program. Today we're going to talk about the general grants program. So big picture, the key accomplishments of our grants program, which has now been active for I guess about 12 years, is that we have to date supported more than $8 million in PWS research. Thank you to all of you who have donated or fundraised or worn t-shirts or whatever for PWS research. That is an amazing accomplishment. More than 125 projects and 75 different investigators. I didn't count the investigators, so that's a little bit of a guess, but I know it's more than 75. And so what has that gotten us, of course, is the question that we all want to know. There's been more than 100 papers published or new advances in PWS research that have been supported by FPWR funds in the medical literature. And we've also really tried to bring new scientists into the PWS field, people who maybe are experts in other fields who can apply their expertise to PWS. And also help young investigators who are just getting started out so that maybe they'll, a lot of times whatever research you get in at the beginning of your career is the research you stay in for the length of your career. So we want to support young investigators so that they will focus on PWS research. The grant program is a place where we can support a broad portfolio, so everything from understanding basic knowledge about PWS to straight up therapeutics development to really practical clinical studies. And I think if you look, we have a list of all our funded projects. You can see the broad portfolio that we've been able to support. Another important outcome of our grant program has been that we have a lot of resources that are now available to the entire community, cell models, animal models, things. And we promote a lot of interaction among the scientists. And I'll just point you to some of the web pages that show what has come of the outcomes of the grant program. So this is just a list of the grants that we supported announced in the spring. And interestingly, as grant cycles go, this tended to have more clinical grants. And here is a list of the new grants that we're supporting currently. And this group tends to be a little bit more animal models and tool development. So I'll take a few minutes and just go through some of those. So we had two grants that were renewals. So we have funded these grants previously. The investigators send a progress report and their plans for an additional year of funding. And in both cases, we felt that these projects were very important. The investigators were making great progress and that it would be worth supporting an additional year. The first one is from Dr. Jeff Sigman, who is an endocrinologist at UT Southwestern, and he is an expert in ghrelin. We know ghrelin is high in PWS. We're not sure exactly what it's doing, and he's done a lot of work to look at what are the possible, what are the broad effects of ghrelin in the normal situation, and what could it be doing in PWS. And so he's done a lot of work to date, understanding how ghrelin impacts many metabolic parameters in a PWS mouse model. He has some very interesting data, and the second year of funding will allow him to complete those studies and draw some what we think will be some very interesting conclusions. So, so we felt like another year of support would be great there. Dr. Tom Scammell is a sleep expert at Harvard. He's a clinical, like Dr. Sigman as well. They're both clinical people, and they have a lab as well. So Dr. Scammell is interested in how some of the important neuropeptides, oxytocin and orexin, impact sleep and wake in PWS, and is again using mouse models to understand that better in PWS. I'll direct you to he gave a great talk at the FPWR meeting in October, and that was recorded and it's on the web, and the link is here. It's an hour long, so but it's worth, to me, it's worth every minute. He's just a very smart guy. He has a real grasp of both the clinical and the basic science side, and he gives an update of the work that he's done to date. And I hope after viewing that, you'll agree that another year's investment is very good there. We have two other projects that are really focused on improving mouse models, and our goal there is to have mouse models that are robust so that we can do good drug development, so that we can know what drugs are likely to work and likely to be safe before we take them into clinical trials. So Dr. Yao at University of Cambridge is looking at one of the mouse models, the SNORD116 PWS mouse model. That mouse model has a limitation because although the mice eat a lot, they never get obese. And it's very hard to study drugs for weight loss in a mouse that is skinny. So so his laboratory has done some studies which are genetic tricks to turn genes on and off at different times during development. And they think they have a way to make a mouse that gets, that starts out skinny but then becomes obese in the SNORD116 background, deletion background. So that will be a real boon to the field. It could be widely shared, and again, it could be used to test drugs. So we want to give them some funding to see if they can, you know, consistently make that animal become obese. A second group is Dr. Tucci and his group in Italy. They're also looking at the SNORD116 mouse, and they've been looking at sleep wake patterns and some other aspects of energy expenditure and metabolism. And we'd like to give him some additional funding to look at those animals under different conditions that might impact their circadian rhythm, their metabolism, and their energy expenditure. So these are two studies that will really help the research side and help build our resources so that we can be more efficient in drug development. We're funding a study by Dr. Nichols. So he has found that there may be a secretion defect in hormones, so that hormones that are made within cells have to get secreted or put out into the bloodstream in order to have their effect. And there's some evidence from his earlier studies that this may not be happening right in PWS. And so he's developed this really clever cellular system, in which he can make the hormones, you know, be red, for example, and then you can quantify, because they're fluorescent, they get spit out of the cells, and you can quantify that process. And that's really important because it will let you, you know, determine definitively if there's a defect in the PWS cells. And if that is underlying some of the problems in PWS, you could then test drugs and really quantify drugs that might be able to improve that process. So it addresses a basic question about what is going on, why are some hormones not being as effective as we think they should be in PWS, or hormones that are made by the person with PWS. And it also could serve as a platform for drug development. We have a project that is really getting at the basic aspects of PWS research, understanding why when a particular set of genes on chromosome 15, the SNORD genes, how come when we lose them, what happens to eventually lead to the PWS characteristics? So Dr. Stam is an RNA biology person. He spends his days really focused on understanding how genes make RNAs and how those RNAs get modified before they make proteins. And he with previous FPWR support, he showed that these SNORD RNAs, there are many of them. We're focused on SNORD116 because it looks to be really important in PWS, but there's other SNORDs elsewhere, like SNORD27. He showed that that has a new role, a previously unappreciated role in how RNA gets spliced. And he showed that the targets of this particular SNORD RNA and how it changes the splicing and how that might modify the proteins that are eventually made by a cell. So now he's going to take those same techniques and apply them to SNORD116 and really look at that gap that we have in our understanding of what happens when you lose SNORD116 to modify the cell to eventually lead to the changes that we see in a person with PWS. And he'll also be looking at, once he defines that, can he engineer some oligonucleotides to replace the function of the SNORD116? So potentially an early approach to genetic therapy. We have one kind of straight up drug development grant that we're going to be supporting, and this is Dr. Roger Cohen, who is an extremely well-known person in the field of obesity and metabolism, has a lot of experience working with drug development. He's at University of Michigan, but has a lot of experience working with pharmaceutical companies as well, and understanding what makes a good drug. And he has a class of drugs that is impacting the MC4R pathway, which is an important pathway in regulating appetite in PWS and potentially disrupted in PWS. And he's going to be developing drugs that will impact that pathway and evaluating them in a PWS mouse model. And the idea here is that these new drugs may be useful on their own, or they may be useful to enhance the action of existing drugs. For example, the Rhythm drug that is currently in clinical trials. Finally, we have a total change in direction, but a really important aspect of the social functioning of young adults with PWS. So Elizabeth Dykins and Elizabeth Roof, as many of you know, have a long history of trying to understand the social implications of PWS and assist not only patients but families in improving their social interaction and reduce stress and sort of make everybody a little bit happier. And there's a real need that they've seen for young adults with PWS. When they get out of the school system, sometimes they become socially isolated. It's a very vulnerable time for depression and mental health issues. And so they want to develop a program that will improve social skills, perceptions, and thinking, and provide a way of interaction. And so they're going to pilot an online study that brings together adolescents and young adults with PWS. And we're excited about this project, which is funded in partnership with FPWR Canada. Thank you, FPWR Canada. We're excited about it because it really addresses a vulnerable population and an underserved, in some ways, demographic. So there's lots of great services when kids are in school, which hopefully we get access to, but there's a bit of a cliff when they're ending up out of school, and especially if they're not around other individuals who, sort of have their same sort of social skills and background. And the long-term goal of this is to develop an intervention that can then be used in multiple settings and kind of spread around the country or around the world to help individuals with PWS be more socially connected and hopefully feel less socially isolated. So we're excited about that as a practical way to improve the lives of those with PWS. Okay, so that's the wrap-up of the grants. Moving into 2017, I think we currently have another set of grants under review that I hope will bring you the results of that review in the early spring. We intend to keep our grant program going robustly because we really are seeing the fruits of that program, and we're excited about that. We view it as a sort of a farm team, so to try out some new ideas, some innovative ideas, and then take the most promising results from the grant program and shuttle them into a more directed research program that allows us to develop new therapies. So 2017, we think, will be another good year for our grant program. So I want to switch gears now and just talk about two pieces of work that have come out recently that just show two different aspects of our grant program and some of the kinds of things that are coming out. The first is a paper that many of you have probably seen on, if you're on Facebook, or in the news. A paper that's coming from Dr. Leibel's lab. Lisa Burnett presented it at the FPWR meeting in October, and I believe Susan will be posting that video. I'm going to present my take on it, but of course Lisa and Dr. Leibel know the work in and out, and so it's probably also worth your while to watch her presentation on many aspects of the work that I'll try to summarize briefly here. So this work comes out of, we've actually supported the Leibel Lab for several years, initially to develop iPS cells for Prader-Willi syndrome. These are cells that they're called induced pluripotent stem cells. Some new technology that came along probably about 5 years ago allowed scientists to take skin cells from individuals, add some genes, do some genetic tricks, and reprogram them so they're stem cells. And all a stem cell is a cell that can then change into lots of different cells. So once the cell is reprogrammed into a stem cell, you essentially add a recipe of growth factors, and you can get them to turn into many different things. You can get them to turn into muscle cells, fat cells, neural cells, pancreatic cells. And the advantage of this is now you can study those cells in a dish. It's sort of affectionately called disease in a dish by the scientific community because, you know, if you want to study the brain of an individual, obviously, you know, there's some ethical issues with taking their brain out to study it, but this allows you to take their skin cells, differentiate them into brain cells, and you can essentially get a read of how those neurons behave in a dish without, you know, without any nasty biopsies. So the first advance for PWS that came after this was a study by Dr. Leibel's lab. We know that hypothalamic neurons, the neurons in the hypothalamus, are likely to be really important in PWS because the hypothalamus is what controls food intake and water intake and emotional control, you know, many aspects that are disrupted in PWS. And up until 2015, people couldn't get those stem cells to look like or behave like hypothalamic neurons. And the Leibel lab published essentially a recipe to turn stem cells into hypothalamic neurons in a dish, which paved the way for the current work that I will be talking about. So they published that, and then they used that technology to look at PWS cells. The cells that they looked at actually come from a patient that was described by Dr. Tauber's lab. This individual has a very tiny deletion compared to the normal deletion. So most of our kids that have a deletion, kids with deletion have a really large deletion that spans this whole region. This particular patient did have most of the characteristics, or almost all of the characteristics of PWS, but had a much smaller deletion. And so that really honed down the important region to this SNORD116 gene. So Dr. Leibel and Lisa and others took cells from this patient and turned them into induced pluripotent stem cells and then made them into the hypothalamic neurons, and then asked the question, how do these neurons differ from neurons from a typical individual? And the way scientists do that is they chop up all the RNA and they ask what is upregulated and what is downregulated. And you can see that in this nice blue and red diagram here. Each line is a different gene that's either down or up. And so, you know, compared to a control. One of the genes, or two of the genes that popped up in this analysis were NHLH2 and PCSK1, which makes the protein PC1. And these were really intriguing to Dr. Leibel and his group because these are genes that are known when they're decreased or mutated in individuals, it causes hyperphagia and obesity. It's a very rare condition, but it causes extreme obesity. So they were really intrigued by the fact that not that they're not completely missing compared to control, but they're very much lower compared to control. As you can see here, and over here, these are the PWS patients, and these are the controls in the black. So they next said, okay, well, what does NHLH2 regulates PCSK1, so I'm not going to talk so much about that. I'm going to talk a little bit about what PC1 normally does, and it's called a proconvertase. So it chops up hormones. Hormones are made usually in a precursor form because they don't want to be sitting in a cell doing their hormone thing. You usually want to wait until it's outside the cell and gets into the bloodstream. So they're made in this precursor form, and they have to be chopped up and processed in order to be functional. So PC1 is one of the proteins that chops up hormones. And this just shows how one of the precursor hormones gets chopped up into a bunch of other hormones that then go off and do many different things. So the team at Columbia then asked, okay, if this PC1 is deficient, then is the hormone processing deficient in PWS individuals? And that's shown here. And you can see, so they compared a control individual to a person with PWS to a patient who had complete PC1 deficiency. And what you can see is there's a subtle change. The person with PWS does not have as extreme a change as a person with complete PC1 deficiency. But when you look at the ratio of a sort of a precursor hormone to the fully processed hormone, which is shown over here, there is an increase in there's an excess of the precursor hormone compared to normal. Again, not as extreme as when you have a complete deficiency, but a subtle difference that over time potentially could have an impact. So they then went back to the SNORD116 deletion mouse, the PWS mouse, where it's easier to get lots of tissues to really look at this carefully. And again, they found the same thing, that the precursor versions of these proteins, these hormones, was higher than it should be, and that ratio of processed to unprocessed was disrupted. Hormones that aren't processed, like growth hormone, didn't seem to be affected, but then hormones that are processed did seem to be affected. So again, supporting the idea that it is this processing that is the difficulty. And you know, that these hormones are actually sitting there, but they're either not getting processed, or, as Dr. Nichols is proposing, they're not getting released, or a combination of the two, and that's what's causing, for example, growth hormone deficiency in PWS. So what are the big picture implications for this? Well, this PC1, you know, it's kind of a central player. It processes a lot of hormones. So if you look at the list over here, it processes hormones that are important in appetite regulation. It processes oxytocin. It processes ghrelin, many, many hormones that regulate appetite, that regulate growth, that regulate emotions and in individuals. So even a subtle change in how those proteins are functioning can have a really global impact. So this is it is a unifying, potentially unifying finding, that there is a decrease in the PC1, and that's affecting many hormonal systems, and that explains potentially why we see all these changes, why we see reduced sex hormones, why we see you know, lower or disrupted oxytocin levels, and you know, has the potential to impact emotion, cognition, and metabolism. So it's a really interesting finding. It's a really exciting finding. I think the sort of the caveats are that there's a long way we still have to go to really show that this is the thing that's causing these problems in PWS. We don't understand yet whether it is the major thing that's causing these problems, whether it's a contributing factor, or, you know, sort of what level of impact does it have on PWS. But it's a great first step, and really opens some exciting opportunities to understand this better, look at new therapeutic targets. So some of the questions we have remaining, you know, the major question is, you know, if we can restore this expression to normal, will we have an impact on the symptoms of PWS? So that's a fundamental question and an obvious next step. You know, so does this pathway account for, you know, most of the features that we're seeing in PWS, many of the features, just some of them? And if we can correct the function of PC1 or make it more active, will we be addressing many features of PWS? We still don't understand exactly how, when we lose SNORD116, we get this decrease in NHLH2 and PC1, and that's important for looking at different, more upstream targets. We don't, you know, there was a whole list of genes that were up or down regulated. So, you know, there's other genes that are disrupted in PWS, and how did those contribute to the picture of PWS characteristics? And what's the best way to try to restore PC1 expression? That's, you know, an obvious and important question. I'll mention that we're continuing to fund the Libel Lab to do additional studies, some of the additional studies. There will no doubt be a lot of interest in the general PWS community to use the tools that they have to address this hypothesis. And Levo Therapeutics, which is a company that Sarah Cotter, a mom of a child with PWS, has recently pulled together, is also partnering with the Libel Lab, specifically on, you know, looking at drugs that might improve the function or activity of PC1. So we hopefully have many bases covered, and, you know, we'll be looking for additional studies to be coming down the line to really solidify, you know, the utility of this target in PWS. And then we have questions about Since it is a protein, potentially, it could be rescued at any time during development, but we don't know that for sure. It may be that there's a developmental component to this that you would have to get expression early on. So a lot of questions left, but really an important discovery that opens up a whole new avenue of research and potentially a whole new avenue of therapeutics. So I'll just take one more minute to mention another paper that I haven't gotten a chance to blog yet, but is also, just so you know that, there's many things going on. We try to highlight some of the papers that are geared towards therapeutics, because we know that's what's of interest to our community, but there's a lot of work going on, including in the area of therapeutics. So I did want to mention a recent paper that came out from Yossi Tam, who's been an FPWR recipient a couple of years back, and has been working very hard with a number of collaborators in the US and Canada and Israel to look at the endocannabinoid system in PWS. So the endocannabinoid system is one of the systems that's important in regulating appetite. And actually, the cannabinoid 1 receptor has been looked at as a target for general obesity. If you remember, there was this drug, rimonabant, some years back that was really very effective, even in the general population, in reducing weight. And there was thought to be some potential in PWS, but unfortunately, in the general population, and even more so in PWS, there was associated psychiatric events. So this rimonabant, the drug, was able to get into the brain, which was thought to be a good thing at the time, and it did cause weight loss, but it also caused some changes in mood, and in PWS, which is a very vulnerable population to psychiatric disorders, it had adverse events. And so Dr. Angulo and colleagues did a small trial with a few patients, and half the patients just had to stop the drug because the psychiatric side effects were so profound. Even though it did look like potentially it was causing weight loss. So that was a real setback to the field, but Dr. Tam and others thought, maybe if you made a version of the drug that couldn't get into the brain, you might still, because this system works outside the brain also. So they thought, well, maybe if we make it where it can't get into the brain, it can still do the metabolic things and wouldn't have the psychiatric side effects. So they've been developing this drug, JD5037, as a peripheral endocannabinoid receptor antagonist. And he and his group first looked in individuals with PWS, shown over here. So this is blood samples from individuals with PWS, and they just asked, is the endocannabinoid system, does it look disrupted compared to normal? And the answer is yes. With that, I'm not going to go into the detail, but you can see that there's a real difference between individuals with PWS and typical control individuals in some of these metabolites and markers of the endocannabinoid system. So that was in humans with PWS. They also looked in mice using the MAGL2 mice, and they also found, as you can see here, that there was a change, especially when the mice were fed a high-fat diet. These mice love a high-fat diet, and it really showed up this change in the endocannabinoid system. So they next asked, they are not quite ready to go into people yet, but you can look in the PWS mouse what happens when we give these mice this drug. And what you see is both in the normal mice, which are shown over here, and the PWS mouse, which are shown on the right, when you give them a high-fat diet, they get heavier, both the PWS and the normal mice. And if you give them a high-fat diet and you give them this drug, they lose weight, and they lose weight quite rapidly. And the PWS mouse even loses weight quicker than the wild-type mouse. So this shows that the endocannabinoid system is disrupted, and this particular drug is really effective at reducing weight. The other thing that they showed is that the MAGL2 mice really, really like the high-fat diet. Could be pretty similar to our kids. that this drug also decreased the amount of food that they were eating. So it decreased the weight, and it decreased the weight because they didn't want as much food. So that's a really promising preclinical finding, or finding in the mice. And this drug is getting ready to go into humans, and so the PWS population looks like a really promising population to test this drug in. So another potential drug to manage hyperphagia and weight loss in PWS that we'll be looking forward to working towards. Okay, just want to wrap up now, remind you of opportunities to participate in PWS research. Please go to our clinical trials page. If your child is anywhere between the ages of 5 and 50, you can really add to our understanding of cognition in PWS and susceptibility to mental illness, as well as behavior in PWS. These are two critically important points. You can do all of these studies online. They're really important studies. They do take this. My son did this study. It takes about an hour. You have to do a lot of cheerleading because it's kind of boring, but it's really important data. So if you have an hour over this break, that would be fabulous. And there are several different studies that Dr. Woodcock is doing for younger children, and you can read about those online and the time commitment to those. But again, everything can be done online. And a reminder, again, I was reminded at the FDA meeting, the importance of the registry, the importance of the data in the registries. Please, if you haven't registered and completed your surveys, take a moment to do that. It's really critical stuff. So I'll just end by thanking everyone and thanking everyone who has provided support either by participating in the registry, advocate reviewing, participating in the research studies, or donating or fundraising. It really, every aspect of it is critical to advancing our mission and making the lives better for all of our children. So with that, I think hopefully Susan will give me some questions to answer.
Thank you so much, Theresa. You always have a brilliant way of really making this research, which is so incredibly difficult for the layman, including myself, to understand tangible. So we are opening this up for questions. Anyone who's on the line, please feel free